Welcome back. Today we're going to be talking about cell sorting. Cell sorting is the process of separating different types of cells from a mixed population. This technique is widely used in research to isolate specific cell types for further study or other applications. There are several methods for cell sorting, but they all involve labeling the cells of interest with a specific marker and then using a device to physically separate them from the rest of the mixture. For example, one method, which is the one we'll talk about today, is called fluorescence-activated cell sorting, or FACS. Uses fluorescent markers, like on standard flow, to label cells, which are then passed through a machine that detects and sorts them based on their fluorescent signals. And aside, cytometrist rant here, please don't use FACS to refer to all flow cytometry. FACS stands for fluorescence-activated cell sorting, not flow cytometry. Another method that you may hear of is called magnetic activated cell sorting, or MACS, and this uses magnetic beads that are coated with antibodies to specifically bind target cells, allowing them to be isolated using a magnet. Today, we're going to be talking about fluorescence activated cell sorting only. Let's take a look. When we look at a cell sorter, you'll find that it has many of the same components as a standard flow cytometer: fluidics, optics, and detectors. However, cell sorters have additional hardware and electronics that allow for sorting. Working our way through the instrument, the cells are taken up and focused in the flow cell using hydrodynamic focusing, all words you've heard before. The major difference here is that most cell analyzers, the cells flow upwards. They're taken up and passed vertically through the flow cell. Whereas in a sorter, the cells are taken up and then loop and go downwards through the flow cell. So that motion. This allows for the cells to be put into a nice downward stream that can be sorted. At the bottom of the flow cell, you'll find something called the nozzle. So let's talk briefly about sorter nozzles. Nozzles are a very important component in cell sorting. The nozzle size is going to determine the diameter of the stream and thus the spacing between the cells, which is important for accurate sorting. As we take a look at these stream images, you can see that the 70 micron stream creates a small droplet. As we move to the bigger nozzle size, you'll see the droplet size increase. And when we reach the 130 micron nozzle, you're basically sorting beach balls. Nozzle clogging, uneven flow, and damage to cells can occur if the nozzle's not properly selected and maintained, and all of these things will affect the outcome of your sorting. Therefore, careful selection and optimization of your nozzle is essential for a successful cell sorting experiment. So choosing the appropriate nozzle size for cell sorting will depend on a number of factors, including the type and size of cells, the sample concentration, the sorting goals, and, well, what's available on your sorter. Some things to think about when choosing your nozzle size. Number one, determine your cell size, or at least an approximate cell size. Like anything else, a nozzle that is 50 microns can't pass a 51 micron cell through it. It will not fit. The nozzle side must be larger than the diameter of the cells being sorted so that the cells can pass through it. As a rule of thumb, you will generally want the nozzle diameter to be at least 4 times greater than the diameter of the cell in order to allow the cells to pass through smoothly. For example, if you're using a 100 micron nozzle, the largest cell should be 25 microns. Smaller, probably better. You also want to think about your cell viability. Nozzle size can really impact your cell viability. Smaller nozzles can generate higher shear forces that may damage cells. Smaller nozzles also run at a higher pressure, which we'll talk about shortly. This can also impact your cells, especially if dealing with sensitive cells. In practice, choosing the optimal nozzle size may require some troubleshooting, some trial and error, and some optimization based on your specific sample. As you can see in the image here, pressure will play a role in this. The smaller nozzles operate at a much higher pressure than the larger nozzles. This is also why you can generally sort a little bit faster on them. So there's an interplay here between the effects of pressure on your cells versus the effects of a longer sort on your cells, and those two go hand in hand and are an important consideration when determining your nozzle size. All of this may affect your cell viability, and at the end of the day, that's what you want to achieve. Cells sorted with a high viability. So playing around with different nozzles may help you improve your post-sort cell viability. And like anything else in flow cytometry, you will need to test to figure out the best solution for your cells. Going back to the fluidics now, another major difference between analyzers and sorters is that the fluid stream in sorters is vibrated by a transducer. This is what makes it cause droplets. If it wasn't vibrated, your cells would just flow out in a solid stream. Adding that vibration to the stream makes those droplets form. This transducer will apply a known frequency and amplitude to the stream. The frequency is then going to determine how many droplets are formed. So for example, a frequency of 80,000 hertz is going to generate 80,000 droplets per second. And then the amplitude is how hard the stream is being vibrated. So if we go back to our schematic here, you can see a point in the stream known as the breakoff point. This is the point where the droplets detach from the stream and form the individual drops, the little packages that we want to sort. This is where the money is at. This point, the point of the last attached droplet, is where the charge is applied to the stream in order for the droplet to be sorted. The distance between the laser interrogation point, so where the lasers hit your cells, and the breakoff point is something known as drop delay. This is the most critical component of cell sorting. The drop delay determines how long the system waits before it applies the charge once a particle is detected. So the cytometer sees your cell up here and knows that in this many droplets, your cell will be here and it needs to be charged to be sorted. That drop delay is critical. If it's inaccurate, the wrong droplet will be charged at the breakoff point and your sort will not be accurately capturing the cells that you think it is. So always pay close attention to your drop delay. If your instrument gives you warning of stream instability or you see stream instability, it's always a good idea to go back and make sure that your drop delay is still accurate in order to maintain an accurate sort. So immediately downstream of the breakoff point, you will find the deflection plates. These are two metal plates, and they work by creating an electric field that deflects cells at a specific trajectory into the collection container, while allowing the non-target cells, the ones that haven't been charged, to pass straight through the instrument and go into the waste. There is an interplay between the charging and the plates that results in the generation of collection streams. So in this diagram, you can see a simple two-way sort, where positively charged droplets are going to the left and negatively charged droplets go to the right. Basically, as they pass through the electric field generated by your deflection plates, the different charge has them move out in different directions. The same idea applies when you have a multiple collection streams. So say for a four-way sort, you would have two streams deflecting one way that would have the same charge, but a different degree of charge to allow that separation of the streams into different collection areas and allowing them to separate from each other. So that is how we are able to get these different populations collected off a sorter. Some sorters can go as high as a six-way sort. Same theory applies here. Just different degrees of charge to allow the streams to separate from one another. All right, we've gotten through the heavy operational stuff. Let's talk about some other important aspects of sorting. First, when you're setting up a sort, there's two important decisions you need to make. The nozzle, which we've already talked about, and the second is what sort mode you want to use. Sort modes can be classified into three major categories: purify, enrich, and single cell. Each of them really live up to their names. It's pretty clear in this case. On a purify mode, you're looking to gain a highly purified population of cells with less of a focus on recovery. How this looks will depend on the sorter you're using. If we look, for example, at the sort modes on my Sony sorter, you can see that within the purify category, there are multiple options for how stringent your sort will be. In these modes, whether or not a droplet is sorted is determined by, first off, a droplet containing your target of interest, and second up, what is in the adjacent droplets, the droplets directly above and below your target cell. As you can see, in ultra purity mode, if any other event is detected in either the droplet before or after the droplet we wish to sort, this droplet will no longer be sorted. It will be aborted because, again, it can't guarantee that it will only take that one cell. So this is where high purity from sorts come from, but at the risk of also lower recovery because you'll generally have a higher abort rate or a higher number of events that are sortable but are not sorted. If we look at the other end, the enriched category, also called yield on some sorters, you can immediately see why the recovery goes up but the purity may go down. In a yield sort, you generally may have multiple droplets sorted in order to ensure that your cell of interest gets sorted. That's what this mode is for, to get as many sortable cells sorted as possible. So this is where you get your higher recovery. However, in this mode, the sorter doesn't care if there are other events in these or adjacent droplets, and that is what gives you a potentially lower purity. And finally, single cell mode. This is really quite self-explanatory. It's looking to put a single cell into a well, generally of a 96 or 384 well plate. This is largely used for cloning purposes, and as you can see is highly selective for the droplet that will be sorted, because you really, in this case, want to ensure only one cell gets into those wells. This is a case where extreme purity is necessary, and because you're generally not capturing a lot of cells, the lower recovery is not an issue. All of these are important considerations when deciding on how you want to sort your cells. If you're, for example, growing up a cell line that is expressing your protein of interest and want a bulk sort with the highest expressing cells to keep in culture, this is likely when you're going to be looking at more recovery than purity. So doing an enrich or a yield-based sort, because over time and after multiple sorts, you can start to weed out those cells that you don't want. If, for example, you're sorting for cells where you're going to be going on a downstream single-cell RNA-seq experiment for a specific cell type, this would be a time where you will want a highly purified population and focus more on purity than recovery. So thinking about the goals of your experiment is really what's going to help you determine what your best sort mode is. Sample concentration is also a really important factor in determining your sort outcomes. A sample that is too concentrated will have an increased number of aborts, resulting in a lower sort efficiency. So when I talk about sort efficiency, what this means is counting or measuring the number of cells that were sorted compared to how many cells could have been sorted. So if someone says their sort efficiency was 80%, that means 80% of the sortable cells were sorted, whereas 20% were lost as aborts. So in order to have a good sort efficiency, the general recommendation is to have one cell every fourth droplet. So run at a cell concentration that allows for that to happen, because in that case, you generally should not have events in adjacent droplets, so you should have fewer aborts, statistically speaking. With a smaller nozzle, because more droplets are generated, you can generally have a higher concentration than a larger nozzle. And remember that little trick I taught you before, where you can calculate the number of droplets generated per second based on your frequency? Well, you can use that if you want to do some math here. You can easily figure out how many droplets you're creating per second, and use this to figure out your cell concentration and what it ideally should be to have one cell every fourth droplet. If you don't want to go to that extent, the easiest thing is to just start with a general concentration, run it on the sorter, see what your abort rate looks like, and adjust upwards or downwards as necessary. So, I talked about what can happen if your cell concentration is too high: more aborts, less sort efficiency. However, if your cell concentration is too low, your sort will generally take longer, and this can also have adverse effects on your cells. Again, like I talked about before, the interplay between speed and time. So playing around with cell concentration is an important part of getting a good sort outcome and for maintaining post-sort cell viability. It allows you to get through your sort as efficiently as possible in as short of a time as possible. And that's really what we're aiming for in a good sort. Sample prep, very similar for what you do for cell analysis, but there's a few tips that I can give you to help you out a little bit here. First and foremost, filter your sample prior to sorting. Not after isolation, not before staining, right before you come to the sorter. While single cell suspensions are important on an analyzer, they're critical on a sorter. In order to sort single cells, you need to give the instrument single cells. In addition to this, cell clumps easily clog a sorter. And when this happens, you're going to have stream stability issues, you're going to have delays in your sort. None of these are good for your sort outcomes. So filtering your sample is the easiest way to avoid this happening. Second up, you've heard me say it before, even more important on a sorter, use a viability dye. Sorting dead cells does no good for anyone. Third up, and along the lines of the concentration theme I was just talking about, count your cells prior to sorting, not prior to staining, when determining your final volume to resuspend your cells in. A fair number of cells can be lost during the staining process, and as we talked about previously, cell concentration is an important part of the sort. So you want to be calculating your cell concentration with the count that is going on to the sorter. Fourth up, EDTA. If you don't know about EDTA, you can check out my video on buffers. I'll link it up in the corner there. EDTA can be used to reduce clumping, and depending on the cells and how they respond to it, this may actually help you keep a single cell suspension during sorting. Again, helping you maintain your single cell sorts, but also helping to prevent clumping. Number 5, adding heapies, again linked in the buffer video, can also be beneficial in sorting. Heapies seem somehow to shore up the cell membrane and keeps them a little bit more robust and protected during the sorting process. And this can help you maintain some post-sort viability. Number 6, collection media. First and foremost, always put collection media in your collection tubes. You would not like to jump off a high dive board into an empty swimming pool. Don't make your cells do the same. Generally, it's best not to collect your cells into media for bulk sorts. Single cell sorts, throw that out the window. You want to collect into media because you have no other way of culturing them otherwise. But in a bulk sort where you're putting a whole bunch of volume into a tube, your cells are coming out of the sorter in a phosphate-based buffer, PBS, and are being sorted, if you're using media, into a carbonate-based buffer. The mixing of these two can cause salt crystallization, and this can poke holes in your cells, resulting in poor cell viability. In our facility, we generally recommend that people sort into straight serum or PBS with a high concentration of serum. Again, this will depend on your particular cell type, so you need to test for yourself what your best collection buffer is. Just always make sure you use one. Number 7, as a general rule of thumb, keeping your cells cool will help maintain viability. Most sorters will have the ability, the option, to have the sort and collection chamber cooled at room temperature, and some even at 37 degrees Celsius. So make sure you choose the setting that's best suited for your cells and your downstream assays. And number 8, the brighter you can make your signal, the cleaner your sort will be. As you can see in this image here, if you try bisect or cut a population in half with a low fluorescence intensity, the downstream collected cells will have a poor post-sort result. They essentially look the same as what you gave the instrument. If we do the exact same thing with a bright population, you can see post-sort we get two really pure and different populations. It's much cleaner and your sort results will be better if you have a brighter signal. Hopefully this gives you the information you need to get started on your sorting project and get started on the right foot. If you have any questions or something about sorting you would like more information about, please leave a comment below. And with that, We'll see you in the next one.